Hello, everyone.
Yeah, let's give it maybe like 30 seconds or a minute and then we can start.
Yep. So I think we have a lot of people. So
Okay, so I'm gonna go ahead and greetings everyone and welcome to another EMN webinar series. My name is Marina Botana and together with my friend Juan Oropeza and the rest of the EMN committee, we are very happy to welcome you all today and glad that you could participate. Before introducing our awesome speakers of today, I want to encourage you all to connect with us and the Metabolomics Society, following the different media platforms, LinkedIn, Twitter, and Blue Sky. And I also want to remind you that this year's Metabolomics Society conference is going to be in Argentina, so for the very first time in South America, in late June, and the deadline for poster abstract submission is May 14th. So now finally moving to our dear speakers directly from New Zealand and Fiji, Professor Rob Keyzers and Professor Andrew Munkacsi, that will be now introduced by Juan.
so it is my pleasure to introduce the speakers. Professor Prof. Keyzers holds a PhD in chemistry from Victoria University of Wellington in New Zealand. He completed his postdoctoral research at Rhodes University in South Africa and at the University of at British Columbia in Canada. He was appointed to the faculty at his alma mater in 2009, where he is currently an associate professor and head of the School of Chemical and Physical Sciences at Victoria University of Wellington. More recently, Rob became associated with the Maurice Wilkins Center for Molecular Biodiscovery and the Academy of Chemical Biology at Ocean University of China. His work combines organic and analytical chemistry techniques to isolate, identify, and characterize biologically active molecules produced by marine organisms, plants, and microorganisms, with the goal of discovering new leads for pharmaceutical, including antibiotics and antifungal agents. Professor Andrew Munkacsi earned his PhD from the University of Minnesota and undertook postdoctoral research at Columbia University Medical Center as an NIH postdoctoral fellow. He was appointed to the faculty at Victoria University of Wellington in 2012, where he is currently a senior lecturer and director of the Chemical Genetics Laboratory. His research integrates genetics, genomics, metabolomics, and chemical biology to investigate rare diseases, drug discovery, and the bioactivity of natural products, with a particular focus on ethnobotanical therapeutics. He has secured over 30 million in research funding and published extensively with Robert Keyzers in leading journals, including PNAS, with work recognized by the Journal of Agricultural and Food Chemistry's Article of the Year Award. Uh, Without further ado, and if you have any questions, please leave them in the Q&A box below, and I leave you to it, professors. I'm gonna stop sharing my screen.
All right. Awesome. Can everybody see my screen?
Yes.
Yes.
Great. Kia ora koutou. Thank you so much for the wonderful introduction. So yes, I'm Rob here in Wellington, New Zealand. And it's a real great pleasure to be able to talk to you today a little bit about some of our work that we've been doing over the last I think it's about 7 or 8 years now, with a local indigenous biotech company run out of New Zealand, Hikurangi Bioactives, looking at the composition and biological activity of a local New Zealand plant, kānuka, which is Kunzea robusta. So Andrew and I are going to split our talk in half. So I'll talk a little bit about some of the analytical platform development that we've done. And then Andrew's going to be talking a bit more about the bioactivity side of things after I've done my bit. So in New Zealand, you may or may not be aware, but in New Zealand we have the Maori people are our indigenous people of New Zealand, and Mataranga Maori is the traditional knowledge system of the Maori. And so Mataranga is a sort of a broad encompassing term, and it's not just about knowledge, but it also encompasses things like worldviews and the spiritual connection that Māori have to the world. And like a lot of Indigenous peoples, Māori feel a very strong connection to the earth and to nature. And this is, Mataranga Māori is very much a system of knowledge that has followed on from, I guess, the migratory patterns of the Māori through the Pacific Islands, and then ending up finally in New Zealand as the final spot of the Polynesian migration. And Mataranga Māori is then passed down through an oral tradition by tohunga, who are sort of the experts, or I guess the academics of Māori society. And as I just said, Mataranga Māori covers a lot of different aspects, cultural, social, but also knowledge base. So one of the key features, and I guess the one that's important for us today, is that there is also a traditional medicine basis in Māori tradition, which is called rongoa Māori. So it's a subset of Mataranga Māori, and that is the traditional medicine of the Māori peoples. Rongoa Māori covers not just the usage of plant materials and other resources, but it's also a much more sort of holistic system of health and well-being. So treatment of ailments through spiritual and mental health aspects are equally important as the I guess the chemical basis that might be come from the usage of a plant material. And so then within Rongoa Raku, that is the subset of Rongoa Māori that relates to the usage of plant materials in their traditional medicines. And these plants themselves are taonga species. Taonga is a term that just means treasured thing, and again that can be a physical thing like a plant, but it could also be a taonga could just also be a concept or a cultural practice. So yes, we have Mataranga Māori, and then within that we have Rongoa Māori as traditional medicine, and then we're talking about Rongoa Raku as the usage of plant materials to treat illness. So kānuka. Kānuka is a member of the Myrtaceae, Kunzea robusta, and kānuka is found all across New Zealand, geographically right from the top of the North Island through to the bottom of the South. There are some interesting differences in the usage of the term kānuka. So different tribes of Māori from different parts of the country call related but possibly different species of plant. They could all be called kānuka, and they all fall under that umbrella term. So there's a little bit of debate as to just what constitutes kānuka as a plant species. Māori used kānuka quite widely in their traditional medicinal applications. It's been used to treat all sorts of infections of the eyes, the mouth, the throat, and for pain relief from inflammation, as well as treating dysentery. And depending on what the application of the plant material was, what the disease they were trying, or the disease indication they were trying to treat, Māori would use the plant material in different ways. So it could be used as a poultice. One of the more common usages, especially for throat infections, was to put plant material into boiling water and then to smell the steam and get exposure to the volatile components that might be present in plant material. Now, more recently, and this is where our collaborators at Hikurangi Bioactives come into the story, part of the interest in kānuka is that clinical trials have shown that incorporating kānuka essential oils into creams are quite efficacious for treating eczema and other skin ailments. And so this has been part of a bigger project that our collaborators have been running to try and find, I guess, a value-added usage for kānuka plant material. This has been part of an ongoing project they've had looking at using endemic plants of New Zealand to re-establish native forests for a variety of reasons. But if you're going to do that, you also need to have some sort of benefit for society, I guess, and that could be economic. So they've been looking at growing more and more kānuka around the country, but then if you've grown it, what do you use it for? And in this case, it looks like it could be a useful ingredient to add into creams for skin ailments. So this has all been led by our collaborators at Hikurangi Bioactives. But as many of you will probably recognize, essential oils, which are the steam distillates of plant material to release the volatile oil components of the plant biomass, because this is from a natural resource, there's probably going to be quite a lot of variation between different plants, be it seasonal or geographic driven differences. And so if we want to be blending in our kānuka essential oil into some sort of cream to treat something, we need to maintain batch-to-batch consistency of our product lines. And so that can only be done if we understand the composition of the essential oil to a very fine level. So in the case of kānuka, kānuka is mainly comprised, or the essential oil, I should say, is mainly comprised of cyclic monoterpenes. So you can see some of the more common components that are found: alpha-pinene, paracymene, terpenine, eucalyptol, or 1,8-cineol. And these numbers here in the brackets indicate the range of percentages that these compounds may contribute to the overall composition of the essential oil. So undoubtedly, alpha-pinene is the major component of the essential oil. Depending on which study you read and how much you trust their analytical platforms, anywhere between 35 up to 70% of the essential oil will be alpha-pinene. But as you can see from these chemical structures, most of these components are monoterpenes and monocyclic. Couple of exceptions, of course: linalool, obviously acyclic, and there is a small component also of sesquiterpenoids as well. So, but certainly the main driver of the chemistry here is very much terpenoid in origin. So if we're going to undertake any kind of wide-ranging assessment of chemical variation to try and tease out some of the seasonal or geographic or even genetic differences, we're going to need to have a platform that satisfies a few different characteristics. Ideally, something that's fast. Certainly, one of the studies we looked at, we were doing data analysis of over 100 samples, and so you need to have enough time to be able to put them through the platform in a reasonable time frame. Obviously, every analytical platform in the world, we want it to have it as sensitive as possible, because this is done for us by students who may have limited experience, as easy as possible is always a good thing as well. Now, because essential oil is derived from the steam distillation of the plant material to release the volatile components, it makes sense, hopefully, that a good start point for analysis would be a GCMS, which of course is a platform designed to deal with volatile components. And so here at Vic, we have a reasonably old GCMS now. It was one of the first pieces of analytical equipment that I got when I was starting out as an academic, so it's over 16 years old now, but it still keeps turning the handle and is pretty bulletproof, so that's good, and it certainly fits that the sensitive and easy platform. But the other thing that is possible is at a more research-intensive institute is that we might have the capacity to use NMR as an analytical platform for analysis as well, using QNMR or quantitative NMR techniques. So what might be the pros and cons, or the advantages and disadvantages of these platforms? GCMS, common and generally quite sensitive, being a mass spec based system, or potentially, for those of you who are familiar, an FID, flame induction detector, and flame ionization detector, sorry, gives quite a high level of sensitivity. In terms of cost, a GCMS is a reasonably inexpensive analytical platform. And I guess the nice thing is that depending on what ionization mode you have, it's quite common to have access to commercial libraries of fragmentation patterns, which will facilitate identification of unknown components within a mixture. And the other nice thing about GC is that the gas chromatograph is very highly resolving, and so you get lovely, nice, discrete peaks for each of the individual chemical components. So it's gives you a very robust and reliable separation for a complex mixture. NMR, the advantages are that it's essentially a universal technique. As long as your compound has hydrogen in it, you should be able to detect it. You also, for unknown compounds, you can use NMR experiments to identify structural components. So if you have an unknown compound, you can start to tease out what it might be. It's a non-destructive technique with a very good dynamic range, and in our hands, very highly accurate quantitation in the molar, or you can get down very easy to nanomolar concentrations quite accurately. So those are the advantages, but there's also disadvantages. Not so much of an issue in this case, but GCMS only works with semi-volatiles or volatile components. Big issue for us is the inability of the machine to accurately differentiate isomers. So compounds that have similar chemical structures, identical molecular formulae, can be very, very difficult to differentiate. We also, and I guess linked to that is we get quite a few false positive identifications. And the commercial libraries, while they're really good, they're not universal. And so if you get some unknown peak coming out on your GCMS trace, it's very hard to figure out what that compound might be. And then with NMR, any of you who've done anything with NMR will know that God never created a less sensitive technique. It is the most insensitive technique known to man. So that's not a good thing for an analytical platform. But it's also a specialist piece of instrumentation. You need highly skilled operators and a lot of infrastructure to run it, and that costs a lot of money. So NMR is a super expensive piece of kit, and yeah, you need dedicated technical support. And in the case of proton NMR, as you'll see in a second, signal overlap does make it difficult to potentially identify individual components within a run. So what our plan was for this sort of pilot study was to compare GCMS and NMR-based analysis of some kānuka essential oils and try and figure out which is the best for our purposes. And we did this on a very small pilot scale. So we used 8 oils from 4 different locations sourced by our collaborators. So that's 2 oils from each of the 4 locations. And then also we throw in 3 commercially available essential oils that we could just buy from the local shop, but we had no idea where they came from or how they were processed or anything like that. So they're essentially unknown samples for comparison. And we just wanted to see, could we get some useful data from these analyses and how the 2 compared. So this is some work that was done by a PhD student at the time, now a staff member here at the university, Natasha Lova. This is the GCMS work that she did. Here you can see early on in our chromatogram, we have our monoterpenes, which are the dominant major components, and then we have a smaller amount of sesquiterpenoid material. This whole analysis takes 32 minutes per sample, but then you have about 8 to 10 minutes cool down and re-equilibration of the column of the oven. So you're talking about a roughly 40-minute turnaround per sample. And we've given you the conditions that we used for our column there. And here we have an internal standard added, which is phenol. Seems to be quite a good standard for us in terms of not overlapping with any of our major peaks. Within this, then here are our 8 sourced oils from our collaborators plus the 3 commercial samples. Across those, we detected 73 individual components. Some we couldn't identify, as you can see here. Others were identified by comparison to their fragmentation patterns and comparing that to both commercially available libraries, but also matching of linear retention index published data to give us a second level of confirmation based off their retention time. And then the ones in bold here, you can see a few of them have been bolded and underlined. Those were ones that we were able to verify with authentic standards as well. And you can see, for instance, alpha-pinene as our major component varies from about 35% there up to about sort of 55% of our oil composition. Some of the other main components, things like paracymene, which is consistent with literature reports. This is our standard proton NMR quantitation. So within this, we were doing this on our 600 megahertz nitrogen cooled NMR system, 64 scans. And one of the most important things when you're doing quantitative NMR is to allow your sample to relax back to equilibrium before you take another scan. So we have a long D1 relaxation of 15 seconds. Overall, this worked out to be 20 minutes per sample. And again, in this case, we used trimethoxybenzene as an internal standard, which gives 2 peaks for the 2 different environments of proton, the aromatic and the methoxy signal. And then if we take the peak area of those 2, we can normalize to what 1 hydrogen of a known concentration would be. And then by comparing to the areas of other peaks within the system, we can relate that to the molar concentration of the different chemicals. So I guess the take-home messages here are that we have more peaks detected here with GCMS, with better peak resolution, but a slower turnaround time. Here we have a faster turnaround time, but less peaks detected due to overlap with the signals. And I'll show you an example in a moment. Down at this more shielded end of the spectrum, where significant peak overlap is a bit of a problem. And here is the, just to show you, here are 11 oil samples stacked one above the other. And just to highlight a few, you can see, for instance, down here, this peak at 7.2 is our solvent, but next door to that, at about 7.1, you can see a peak that does vary across them. And there's other peaks that increase or decrease depending on the provenance of the oil system. So we can use both platforms to do the analysis then, and that works well. The next thing that Natasha did was to then do a little bit of statistics. And the first thing she looked at was what was the correlation between the two platforms. So in our NMR, we detected pinene, paracyamine, terpenine, terpinolene, terpineol, linalool, and viridiafluorol. And the first thing to note is that actually the correlation between the two platforms for pinene, the major component, was actually pretty rubbish. We didn't get a very good correlation at all, and certainly not with any strength, statistical strength, which was somewhat surprising to us. However, most of the other components we actually got very good correlation to, with some strong statistical basis that the two platforms are essentially giving us the same trends at least. So that gives us some confidence that overall the platforms are telling us roughly the same story, and that either of them can be used to screen our oil samples. Natasha also did a PCA of the two different platforms. And I guess first things to note is that they give similar levels of discrimination. Both give roughly 50% discrimination across the two platforms. And the other nice thing is that largely the groupings hold. So for instance, oils 5 and 6 are diametrically opposed to oils 1 and 2 under both platforms. And as would probably be reasonably expected, the commercial samples, K1, 2, and 3, which we don't really understand anything about their provenance, are just distributed across the PCA, and so that we wouldn't expect them necessarily to cluster with any of our other oil samples. So overall, the GCMS and the NMR platforms are giving us the same story. Now, that work was published a couple of years ago in phytochemical letters, but more recently, we've taken this a little bit further. So proton NMR is quick and easy for qNMR, for quantitative analysis. And for those of you who are familiar with NMR, you'll know that proton, 1H, is the most sensitive nuclei available for NMR analysis. However, as I've alluded to, for a variety of reasons, proton spectra do have signal overlap, which makes differentiating individual compounds quite tricky. So here's an example where we have some nice discrimination of our kānuka samples at the more deshielded ends from alkenes and aromatics, but in the more shielded end of the spectrum, there's a significant signal overlap, which makes differentiating individual signals from individual components quite tricky. Now, organic molecules, if you think about the origin of the term organic, what that means is it's got carbon in it. Now, carbon is ubiquitous then in organic compounds, but unfortunately, due to the selection rules for NMR active nuclei, carbon-12 is NMR silent. It does not respond to a magnetic field and cannot be detected by NMR as a platform. Only carbon-13 is. And the problem with carbon-13 is it only corresponds to 1.1% of carbon in the environment. Not only that, but there's something called the gyromagnetic ratio, which is the property, the inherent property of a nucleus that dictates how sensitive it is in a magnetic environment to give an NMR signal. And because carbon-14's gyromagnetic ratio is a quarter that of proton, it's only a quarter as sensitive as proton. So once you've taken into account both the low percentage abundance and the low gyromagnetic ratio, carbon-13 tends to be about 5,000 times less sensitive than proton for NMR analyses. Then, for technical reasons, it's also hard to get an accurate area measurement under a carbon-13 signal that you could then use to relate to concentration. You can get around that using some experimental changes, but by and large, a standard carbon spectrum can't be really used in a quantitative sense, especially when it's a mixture. But the good thing with carbon is that you get a carbon spectrum that gives much sharper lines, and there is much less signal overlap. So you get one peak per carbon, nice and tall and sharp, and it's very unusual to get overlap. So if you have complicated mixtures, this makes it much easier to differentiate the signals in a complicated mixture. Now, the nice thing with the kānuka essential oils is that we're essentially not sample limited. This is not a study where we're looking for biomarkers in urine or something like that, where you've got a very limited sample set. Here we have each batch is generating milliliters of essential oil. And so we could largely take as much as we want to throw it into the machine. So we can get over the inherent lack of sensitivity of carbon NMR by just adding in more sample. So we've had a go now at doing quantitative NMR, but with carbon-13 as our platform. So this has been done by our NMR technician, Mr. Juan Forster. And here at the top is just a standard carbon spectrum for alpha-pinene. This was a 30-minute run, 12-minute run, I should say. And you can see that the peak heights and therefore areas as well for all of these signals vary. And the main reason for that is the lack of full relaxation back to equilibrium. So alpha-pinene, if you were just to run a standard spectrum, you wouldn't be able to accurately quantitate based off these peaks because they're not all giving the same amount of signal per carbon. But our first go at turning this into a quantitative analysis on our 600 stronger magnetic system with a much longer D1, so going from 2 seconds up to 126 seconds. Now, all of a sudden, our peaks are largely giving rise to the same area, and irrespective of which carbon they are in the molecule. Now, the problem, hopefully, that you may have noticed is that the total time is 204 minutes. So we're getting up to almost 4 hours per sample, which is very not fast for a rapid platform. But this was our first go at this. But we did have a go with this 4-hour analysis, and here is our on 3 oils. Again, this is alpha-pinene at the top, and then our 3 oil samples. And again, you hopefully can see that alpha-pinene does come up as the main component, and it's major component as we would expect. But there is some variation between our samples. And if we zoom in, there are some minor variations in different components, and we can still actually detect some very low-level metabolites accurately, even with this insensitive technique. So we're now moving towards a more sensitive, optimized approach. So at the moment, our initial parameters here, where we had that almost 4 hours per sample, there was 50 microliters of oil made up to 750 microliters in solvent. Now we're down to only 64 scans instead of 128, a relaxation of 60 seconds rather than 125. We're still getting a reasonably big noise level, but the amounts are reasonably comparable. I guess the sensitivity here is reasonably comparable between those two approaches. And the good thing is we can still add in more oil to increase the sensitivity. So we're having a go now with more like 100 microliters of oil. So that's about 84 milligrams or so of oil into our chloroform. And we should be able to get this down to about 30 minutes per sample and still maintain adequate levels of sensitivity. So even though carbon-13 is an insensitive technique where you are not limited by sample, this becomes a viable option And as you can see here, hopefully much better likelihood of being able to differentiate minor components and not have those signal overlaps that you have a problem with in Proton. So by conclusion, you've heard enough from me. We have generally good correlation between GCMS and Proton. In our hands, Proton NMR is faster and easier, but you're less able to differentiate metabolites. And GCMS gives you a more comprehensive platform, overview of what's in there. Interestingly enough, the large driver of differentiation is not actually the alpha pinene, it's other components. And at the moment, as I just showed you, we're working towards using carbon-13 as our next attempt to assess kānuka essential oils. So I just want to quickly thank Natasha and, I didn't add Jan's name here. Oh, my bad. So Jan Vos is our NMR technician. He should be here as well. And then couldn't do this without the assistance of Hikarangi Bioactives, who have supplied us with funding and also all of the samples as well. I'm going to stop there and hand over to Andrew.
Please remember to unmute yourself, Andrew. We can't hear you.
Hi, everybody. Kia ora, ula. Thank you for giving us the opportunity to present our research. So I'm just going to continue on with this metabolomics guided bioactivity studies on the kānuka project. And our lab takes advantage of yeast as a model system. We use robotic plating of genome-wide mutant libraries. So you can imagine libraries of strains where each strain is missing a different gene, or each strain has a GFP fused to it. So then the protein fluoresces, and you can monitor that with a microscope. So we have this kind of robot here where the plate on the left will contain yeast strains, and the robot will pick up a pad that essentially is like a fingerprint in a grid format, in 96, 384, or 1536 format, depending on how many yeast are on each plate. Then it will pin down, touch the yeast from the source plate, and then transfer it to a new plate that, say, has some kānuka oil in it. And you can compare growth of the yeast strains on kānuka oil versus the control. So for example, here you might see here this plate contains kānuka extract, whereas the plate on the left is the control. And see this colony here, everything's done in quadruplicate. So the gene that's missing in this coordinate, it grows when there's no plant extract, no kānuka oil extract, it grows perfectly fine. So that gene is not that important. But the gene is very important in terms of responding to the plant extract. So it's not to say that that gene is the target, but it's just involved somewhere in the metabolism of the plant extract. So you can think of it as like that gene is missing, the yeast is not growing as well, therefore the gene is somehow required for the cell to buffer and deal with that extract. We can also use high throughput confocal microscopy to look at the abundance and localization of proteins. So we would have a high throughput microscope here. Essentially, we would do the same thing. The robot would pin the libraries, but instead of onto agar, it would pin them onto 384 well plates that have the black walls, and the microscope would find the cells, take lots of few images, few fields per well, and we can track the localization of the GFP tag proteins and the abundance in the treated cells compared to the control. And we have a machine learning platform that does this using these hallmark proteins that are known to localize to certain components, and so we can train the analytical tool to identify where the GFP tag protein is localized to, and also quantify the abundance. So we'll talk only about the kānuka as a sustainable resource in drug discovery. Rob gave a great introduction into the rationale of this. These people are on the board of directors for our collaborator, the Hikarangi Bioactives, and their main reason, one of their reasons, is to develop to find some added value to kānuka, and they've made incredible progress in terms of anti-acne and anti-eczema. But there's also an environmental component in that the pine forestry industry exists is quite prominent there in the East Coast. And what happens is the pine doesn't handle the change that's been happening in the climate so well. So there's a lot of erosion, and you end up with houses like this, or even on the beach, just thousands and thousands of logs because the pine plantations are washed away. So kānuka is much more better, being a native plant, has a stronger root system. So I'm just going to cover 3 metabolomic guided stories about kānuka oil bioactivity, starting with antifungal synergy with rapamycin. So we did that gene deletion analysis, and we were just comparing growth and identifying which genes are required for the response to kānuka extract. And we've done this for many compounds, and other people around the world have also done it for many compounds. So there's libraries of these chemical genetic profiles for thousands of compounds. And one compound where there was some striking similarity was rapamycin. And so in this visual here, this is just each dotted line area on this map are where the genes would go for particular fundamental biological processes. So there's 17 of these delimited areas for 17 very important fundamental processes. And you can see that there are some similarities between kānuka extract and rapamycin. So that got us wondering whether kānuka was doing the same thing as rapamycin, or possibly whether it could be synergistic with rapamycin, rapamycin being so well investigated as an antifungal, but now recently developed and FDA approved as an immunosuppressant, and also being looked at for almost any bioactivity, really, because it anti-cancer, anti-aging, so lifespan extension, so many uses, potential uses, but FDA approved as an immunosuppressant. So we looked initially at the synergy, and this is just in yeast. So this is looking at percent growth, a ratio of treated versus untreated. So if the ratio is 100, that means there's no difference between the treated and the untreated. And so we use these very low amounts of rapamycin and very low amounts of the kānuka steam extract, where those amounts basically have no impact on growth. But when using those two same amounts together, there's about a 50% reduction in viability in the yeast. So there's clear synergy, at least in terms of inhibiting the growth of yeast. To get a better sense of how that synergy is happening, we looked at the gene deletion library, and we saw that there was a handful or a few almost 2 handfuls of genes. You can see when both compounds when the steam extract and the rapamycin are used, there's quite a bit of sensitivity to the dual treatment as compared to when there's just treatment of one or the other. Those genes, of course, to me, as well as other yeast geneticists, not every yeast geneticist knows every single name. The yeast genes are typically 3 letters and a number. So just as to you metabolomics people who don't work in yeast, it's just 3 letters and a number. What does it mean? So we really wanted to put it into a functional context. So we did a network analysis where we included those genes that were on the previous slide, and their interactors, so other genes that those genes interact with, to get a broader perspective. And so you can see here in the center is the target of rapamycin. So that's the actual physical target of what rapamycin binds to. And so through this analysis, we're able to at least suggest that the mechanism of action might just be increased rapamycin effect, and as compared to another effect happening on the cell. So there's some future work that still needs to be looked at to get a better sense of that mechanism of action of this driving the synergy. Autophagy is a major process that's induced by rapamycin, and we recover some of the same. We cover enrichment for some of the same processes that are that happen with rapamycin alone. So it is possible that we're seeing the rapamycin effect, but by using less rapamycin along with the kānuka oil. The dual treatment of kānuka steam extract and rapamycin increases the number of vacuoles that a certain protein gets sorted into during endocytosis, which is consistent with what would happen in autophagy. Likewise, there's a reduction in the number of reactive oxygen species with the dual treatment. So each flowing cell would be a cell that is that contains reactive oxygen species, so has an oxidative effect, whereas there might be an antioxidant effect of the dual treatment. And interestingly, each treatment alone does have an antioxidant effect, but it really does it might be even further increased with the dual treatment. Then with the help of Rob and student Storm, we were able to do some bioactivity guided fractionation to identify the key compound that's driving that synergy. So essentially fractionating based on polarity, and for each fraction, seeing if it's synergistic with rapamycin. And fraction 2, you can see is with rapamycin is giving the same kind of growth inhibition as the steam extract and rapamycin, and that fraction 2 was identified to be neurolidal. So there's some pretty interesting things to pursue here, given the massive interest in rapamycin for all these different components. Rapamycin having compound being discovered from soil in Rapa Nui, and unfortunately, no benefits have been shared with the indigenous people of Rapa Nui. So it's our collaborators at Hikurangi Bioactives that are keen to set up benefit sharing if the synergy is further developed and even commercialized, then our collaborators are keen to set up a benefit sharing with the people of Rapa Nui. The next story is about anti-acne activity, and so Rob mentioned the eczema cream that's being developed, and then the oil is being produced, and then from different locations around New Zealand, and then being shipped to the U.S. For production of the cream that then gets exported, and all the landowners share the benefits. So that's the working model for the oil production and the benefit sharing. One thing that we wanted to look at was the seasonal variation. So we're trying to get a sense of when to collect that's best for the anti-acne activity. So we looked at the growth of a bacterium that's acne associated. That's called Propionibacterium acnes. And so we looked at the growth of this bacterium in response to 90 isolates of the kānuka oil, of the kānuka steam extract, from one season, which was summer, and another season, which was spring. And these samples, what you can see generally is that there was variation depending on whether it's from this land block, or this person's land, or this person's land, or this person's land. There's variation, as you would expect from nature, but the upside is that there's always strong inhibition. So almost always more than half inhibition of this bacterium. To get a sense of what the key compounds are, we did partial least squares regression analysis, where we mapped out how well each compound correlates with the bioactivity. And there were a bunch of compounds that were identified as being statistically significantly correlated with the bioactivity. But at the current time, we haven't looked at any of these on an individual basis. So that would be good to do, to really see if the anti-acne activity can be achieved just with limonene alone, or any of these alone. But interestingly, alpha-pinene, the major component, is not predicted to be important for the bioactivity, but it definitely is important for confirming that you have a high percentage of that alpha-pinene for quality control of the eczema cream. Lastly, some unpublished work that we've shown kānuka oil regulates lipid droplet metabolism. So lipid droplet metabolism is integral to health and disease. So lipid droplets are these little things they're double membranes, organelles in cells. They were named organelles a couple decades ago. And they're key components for energy storage and for storing fat as well. So phosphatidic acid gets converted to diacylglycerol via a couple different genes in yeast, one of them being PAH1, phosphatidic acid hydrolase 1. DAG gets converted to TAG, and then TAG, along with steryl esters, get packaged into these lipid droplets. So lipid droplets are mainly composed of TAG and steryl ester. So what was quite interesting was that in a mutant PAH1, that only, you can see here, if this gene is lacking, then you won't have production of DAG. You'll have less TAG, and you'd have less lipid droplets. So there's really a lipid droplet deficiency when PAH1 is missing. But interestingly, when the kānuka oil is treated to these cells, the lipid droplets get bigger and increase in number. the kānuka oil, the steam extract reduces protein levels of proteins that are involved in tag metabolism. There's a few shown here. So here you have the control. You're just looking at the GFP intensity, and you can see examples like this DGK1. You can see that the GFP intensity is pretty good here, but it's going to be less in the kānuka steam extract treated. So that led us to look at overexpression of DGK1 as a means to revert the effect of the kānuka steam extract on lipid droplet abundance. So in this case, you've got the control and the kānuka steam extract treated, but each in wild type or PAH1, you're overexpressing DGK1, and so when DGK1 is overexpressed, you don't see that lipid droplet accumulation. Interestingly, when we did the bioactivity guided fractionation, we ended up with the most common and most abundant component, and that's alpha-pinene. So here with alpha-pinene alone, you can see you increase the lipid droplet number just like you do with the kānuka steam extract compared to the control in that PAH1 mutant. So everything is specific to this PAH1 deletion mutant. So that led us to this working model for how the kānuka steam extract might be increasing lipid droplet biogenesis in this one specific mutant. And so when this gene is deleted, then what you might have is the extract inhibiting DGK1, which counters the effect of the PAH1, and that might be how you get the increased lipid droplet formation. So in summary, these three metabolomic guided bioactivity stories, we've shown antifungal synergy with rapamycin. So we have nice clean results in yeast, but it's not yet known if there's less toxicity than rapamycin alone. And that would be particularly interesting to work out with in mammalian cells. The anti-acne activity, this bioactivity is considered across 2 seasons, so that's useful for the commercialization part of the project. The key compounds have been predicted with statistics, but not yet experimentally tested. And I think and as far as the regulation of lipid droplet metabolism, what's really interesting about that is that unlike the antimicrobial or anti-acne activity, which was informed by traditional knowledge, the regulation of lipid droplet metabolism was not informed by traditional knowledge. And so what would be interesting to do is a lipidomic analysis of this bioactivity to get a better sense of the mechanism of action, see if which lipids are going up or down. And importantly, I think this is potentially limited to cells with specific lipid metabolism defects. So yeah, the work could not have been done without everyone at Hikurangi Bioactives, and we've also had good support, which is an initiative of the kānuka and the Maurice Wilkins Center and our Center for Biodiscovery at our university. So thank you everyone for this opportunity. I look forward to talking about the questions.
Thank you very much, Andrew and Rob, for the amazing talks, and thanks for joining us from your vacation time in Fiji, Andrew. The connection was a bit delayed,
I'm sorry.
but
Yeah.
no, it was great. It was a great talk. So we do have a few questions on the Q&A, but I do have a question to start with. So super interesting, the research very focused on kānuka. But is there anything else happening with other New Zealand natives? Because there's such a great diversity of plants that are part of Rongoa and why the particular focus on kānuka?
Yes.
Go ahead, Rob.
No, you go.
No, you're in a better connection. Go ahead.
I was going to say, yes, we have been looking at other plant sources as well, but I think part of this project was largely driven by that aspect relating to soil erosion and stability of land that Andrew referred to before. Kānuka grows really, really well in the geographic area where our collaborators come from, and they were actually looking at trying to stabilize land in a world where there's more, with global warming, when there's more flooding and things like that. How do we actually protect people from the land and all these pine trees flowing down rivers and knocking over buildings? So one of the best plants to stabilize the land is kānuka. But if you're going to grow so much kānuka, what are you going to do with all of it? And that's where, right, let's look for a value add to that from that biomass. But that isn't to say you're absolutely right. There's plenty of other plant materials that form part of rongoraku, and we are looking at those as well.
Super interesting. I think we can, thank you so much. We can move on to the chat now because we have quite a few questions. I'm going to read the first one, which is someone new to GCMS, and he or she would like to know about the linear retention index. And what would be the range of confidence when comparing the theoretical and the experimental linear retention index values?
Fantastic question. So LRI is very useful to compare results generated on different machines. I guess the most important thing is that it has to be the same column architecture, the same stationary phase, and done with the same analytical standards. So in our case, we use the N-alkane series for our LRIs. People debate what's a suitable variation. I've seen anything up to 30 to 50 LRI units as being okay, and we typically use 50. There's enough instrumental variation just within our own machine, sample to sample, that if you went anything lower than 30, you probably wouldn't call the same peak the same thing run to run, and that's pretty standard on most GCs. So yeah, long answer for a short question. I'd say somewhere between 30 and 50 is okay.
Awesome. I'll go ahead with the next question. It says, do the kānuka oils vary from geographical origin or environmental conditions?
Yes, is the answer. So we've done some studies with different land blocks of kānuka. So the study that generated a lot of the data that Andrew talked about came from about 45 samples or so each in 2 different seasons. So we ended up, or maybe even more, close to 50 samples across land blocks in 2 seasons. So we ended up with about 100 samples altogether. And they do vary based on which land area they come from. And there was also seasonal variation in some components as well. And that's, I guess, you kind of expect that. Is that based on the geology, of the plant, or is it the genetics? That's a big question and an unanswered one at this stage. As I alluded to right at the start, there's still some confusion, I guess, around just what is kānuka and how many different variants and subtypes there are. So the genetic variation could be the main driver, or it could be the geographic. And then there have been some other studies done by other researchers in New Zealand looking at geographic variation, and it can be wildly different. To the point where actually one study actually reported a kānuka sample that supposedly had no alpha-pinene in it. Now, considering every other study says that's 35 to 70%, chances are they just identified their plant wrong and it wasn't kānuka at all. But there has been lots of reported variation. Oh, Marina, you're on mute.
Sorry. I have a quick follow-up question on that because since there's some seasonality variation on the composition of the oil as well, what would be, the best season for the harvest based on like if we want to target acquiring more bioactive compounds, would there be an optimum time of the year for the sampling, or have you looked at that at all?
Yes, so we did look at it. Andrew, can you remember which seasons we collected in?
We're summer and spring. So just 2 seasons. So we didn't collect in winter. And just for people to know, internationally, New Zealand is not as warm as you think it is. So winter is close to a real winter. It just doesn't get snow. It's close to cold enough. So it would be quite interesting to look at winter, see how the metabolites, see that metabolomic profile in a season where really the plant's not growing. It would also be good to look at autumn. Yeah.
The other thing is there was variation in the chemical components though. So when we looked at the bioactivity plot, the partially square regression that Andrew showed, and some components are more associated with anti-acne and others. We looked at MRSA and others. Once we've tested those compounds and shown that they are efficacious, we could pick the right season to maximize those compounds. So yes, there is, but it differs depending on which bioactivity we were chasing and which components were the main driver.
Yeah, because there's a question in the chat asking if the metabolomics findings align with the therapeutic uses described by indigenous knowledge. And I wonder if Rongoa would actually recommend a season for the harvest, or.
I think that's where Rob could comment quite good here. Yeah, not with kānuka, but he's done some amazing studies on kawa, and looking at the traditional knowledge and them informing when during the day is best to harvest, as far as when the light is hitting the leaves, and that's an incredible variation just within a day.
Yeah, so we've had I had a student who worked on a Maori student who used her own family traditional knowledge around when is the right time of day to pick the kawa leaves to have this effect versus that effect, and for instance, yes, light versus shaded parts of the tree have different effects, and that was borne out with variation in chemical composition. So that's unpublished work, unfortunately, because it's private knowledge of the family. They haven't wanted that to be released into the public domain, but very interesting study. I'm gonna hit it back to Andrew. Nice sort of forehand strike about the therapeutic uses by indigenous knowledge though for kānuka. I think there's 2 answers there.
Yeah, so we've looked at a few bioactivities, so 2 antimicrobial bioactivities that would be consistent with the traditional knowledge. So that would be antifungal activity, and not just against Saccharomyces cerevisiae, but against Candida species that are pathogenic to humans. And yeah, there is definitely antifungal activity there. There's also antibacterial activity, which would be consistent with traditional knowledge. And for that, we've looked at standard strains of E. coli as well as pathogenic and drug-resistant strains of bacteria like MRSA. And there is antibacterial activity. We haven't looked, and then there's the clinical trial, of course, was done for anti-acne and anti-eczema. But other than that, we have not looked at other bioactivities. So like, yeah, and then some of those are just more elaborate and out of safety zone. Is looking at you'd have to look at mouse models.
So we saw a lot of NMR characterization. There's a question in the chat that says, have you acquired any data from untargeted LCMS of kānuka?
The short answer is no. So because of the way, and this actually to some extent I think answers the next question as well. These are essential oils which are generated by steam distillation of the plant material, which then carries across the very volatile components, which are highly non-polar. And so they don't typically interface particularly well with reverse phase chromatography, electrospray ionization. So your standard platforms for general untargeted LCMS, we did have a go, but we just didn't get any good results. And that's not to say that you can't do those sorts of analyses, but our instrument just isn't set up for those less polar materials. So had a go, didn't work, basically, is the short answer for that one.
Cool. I'm going to move to the next question from Jonine Seo, saying, thank you very much for the interesting talk. And two questions. The first one is, are there any changes in the contents of bioactive compounds of kānuka extracts, depending on the extraction method? If so, what extraction method did you end up using? And what are the contents of those compounds in the extract as well?
Yep. So as mentioned, this is a steam distillation extraction methodology. We don't actually do that. Our collaborators are collecting the kānuka in their local area, and part of the idea is to create a industry involving as many people in the community as possible. So they do the steam distillations for us in Ruatoria, and then send us the essential oils. So that's the extraction methodology. And yes, there is variation depending on how you form the steam distillation. For instance, if you go for longer, you'll start to extract less volatile components. So there's another class of metabolite called triketones. They're not terpenoid in origin, they're polyketide related compounds, but they're not that volatile. But the longer you leave the distillation, the more likely you are to volatilize some of those triketones into the steam distillation. So our collaborators have spent some time optimizing their steam distillation to get the terpenoid components, and then they stop, and then that is what they send us. And that gives us a little bit of control, actually quite a lot of control on the consistency of the extraction methodology.
Thank you. And he follows up as well, asking if those bioactive compounds in kānuka extract are actually present in the systemic blood when they are taken orally, or how much is absorbed to systemic blood? Are they metabolically stable? So have you maybe analyzed any plasma samples of patients or?
No, we've never done that. So unfortunately, we can't answer that question, I'm afraid.
For future work.
Exactly. Yep. And remember that also most of our work is focused on topical application, just because that's a much more a low barrier when it comes to things like clinical trials. As soon as you start giving stuff to people to consume orally, obviously the barriers to safety is much higher than if it's just something you rub on your skin.
I have a question myself. So, you seem to work a lot with the industry, right? With the company that is producing the oil. How do you structure your collaboration with them? Do they propose applications that you then investigate, or you do the research process like joint work or something like that?
You want to take that one, Andrew, or do you want me to?
Yeah, that's a good question. The entire collaboration has been was initiated by our collaborators. They were aware of the facilities we had, and they had particular questions in mind, and they asked if we were interested in working with them and working with students to do this kind of work. And so yeah, in a way, it's their questions that drive the work.
Awesome, thank you. The next question says that New Zealand natives and the unknown interactions with the soil microbiome seem so awesome for the discovery of new molecules. Have you found any other rapamycin-like bioactive molecules?
So not rapamycin related molecules as such, but we do have other groups within our university that we work together with. I like my sponges and marine stuff, but we have other soil related researchers looking at the soil microbiome and also doing genomics guided discovery of biosynthetic gene clusters. So we're definitely looking at new type 1, type 2 polyketoids, RIPs, NRPSs, the whole suite of other biosynthetic classes. So no new rapamycins yet, but you never know what we might find.
Awesome. So I'll follow up with another NMR question from Andrew Lewis. Have you tried NMR relaxations agents to speed up the carbon-13 QNMR? CR ACAC recycle delay could be reduced to 1 to 2 or less seconds, and why not HSQC NMR? I'm not an NMR person, so I might be missing out something here. I'm sorry.
No, you did very well, Marina. Excellent question, Andrew. We should talk more. No, we haven't tried relaxation agents, but that's an excellent idea. I guess one of the things we were trying to do was to simplify the approach as much as possible, so that we just take oil, squirt into chloroform, chuck into the machine, and measure with as minimal interaction and intervention as possible. But certainly wouldn't hurt to make up a stock solution of chloroform with chromium macaque and chuck it in and see, have a go. Why not HSQC? Just haven't got there really. I've never had much luck trying to do, I guess, volume concentration measurement on a 2D plot. I know you can, just we've never had much joy with that. So no reason not to try it. We just haven't.
There's another question asking if there were any integration between the results from the GCMS NMR and the gene approaches.
I guess if I'm interpreting that question correctly, so I guess yes, to some extent, when we're looking at which of the bioactive components in the steam distillates, and then relating that to the bioactivity, especially I guess the antifungal bioactivity in relation also to the rapamycin story. But anything you want to add, Andrew?
Yeah, we haven't done any full, proper, complete analysis to try and correlate the chemical genetic profiles and the actual metabolites detected. That's a really good idea. Thanks for that.
And I'm gonna move to our final question in the chat, which is from Sarah Hafez Gohan. And she thanks you both, and it was a really nice talk. And she's interested in NMR metabolomics and asking if you could find some interesting phytochemicals that are less studied.
Excellent question, and we're always looking for new interesting compounds. The reality is that certainly in kānuka, most of because it's so well studied, and plant metabolism within the Myrtaceae is so well understood, there is a lot of commonality in the compounds across geographical areas, the globe, right? And so most of the compounds we're finding are just very simple, well-known metabolites, which isn't to say that we might not have some interesting compounds. And certainly in the GCMS analyses, roughly half, I think, of the compounds were ones that certainly we couldn't identify based off mass spec fragmentation patterns. So I suspect there is definitely some very low abundance new compounds in there. but we just haven't had the time to chase those down. Takes time. That's the reality. You need people to do it.
You need technical development effort. Yeah, it takes a whole team, right?
Absolutely.
Yeah, so thank you, Rob and Andrew, very much for the super interesting webinar today and for everyone who joined us live. I look forward to seeing more metabolomics research in New Zealand and to promote and share that with the international community. It was an interesting and inspiring talk from you both. And for the ones who missed part of the webinar and those who couldn't see it today live, we know it's tricky with the time zones, but don't worry because the recording will be soon available in the Metabolomics Society website. And feel free to reach out to the EMN committee and to MedSoc whenever. And thank you all again very much.
Thank
you for
you,
coming.
everyone.
Thank you, everyone.
Bye-bye. See you guys. Thank
Bye-bye.
you.
Bye-bye.